Well, good afternoon, everyone. Thank you for the opportunity of being here for the first time in this arena for me. And thank you for still being here in this last talk for the session and not being outside enjoying the sun. I have to say that after Emmanuel's presentation this morning, well, the beginning of this session, I felt a bit embarrassed because I would expect to bring to you much more final results that I have. So face it as a concept, as really preliminary data, and you will see why during the presentation, why 3 or 4 months ago when I wrote the abstract, I thought that I would be much further now than I am. So that you will, you will find out soon. I have a bit of background slides, which are basically, I think, mainly meant for me, because you are all pretty well aware of all of these, but it's a way for me to keep the track on what is really important and not get distracted with evolutionary or ecological peculiarities and questions, which is usually where I get lost. So basically, in all the processes of colonization, of domestication, there's the population suffers several bottlenecks, and there is, with that, a lot of loss of genetic variability. Nothing new here, and if we would take a snapshot in time, that is not even that relevant. The problem is that biological systems do evolve, so which means that variables always change, and history always matters. And that's why this loss of variability is indeed a problem. Because it reduces the ability of the species to evolve in response to future changes in the environment. And that's the only thing that we have for certain, is that change is ahead, and this is a world in constant change. And now we are even more aware of that with all the global changes that our planet is suffering, in our awareness of that. And this narrowing down of the genetic variability among the cultivars has real implications for survival from several biotic and abiotic stresses. So, you know this again better than I, the success of a crop improvement program depends definitely on having sufficient genetic variability, so you do need to look for it. And one of the strategies that have been talked here over and over is to go on cross-compatible wild types and find desirable traits in their genes. And here became my question, shall we just go randomly to gene banks? Shall we just, how do we do this? And I started thinking, would it be possible to have a simple and relatively fast tool to try to find habitat-adapted wild genotypes? I mean, can we find a correlation between wild genotypes and local environments? Because then we could go into those populations that our model predicted as having more interest, and then yes, then with other techniques like gene by sequencing approach that Shelby had talked yesterday, or all the other traditional methods of testing and growing in laboratory experiments, and really look for the genes of interest. So that was my back of my mind thought, and for that we selected a specific gene, the alternative oxidase, because it's known to reduce the level of oxidative stress, and therefore it's not responsible for a specific stress as far as we know, but has evolved in quite a lot of stress responses. Just in short, AOX is a protein that is located in the inner membrane of the mitochondria, is related with growth and development, responds to stress conditions, both biotic and abiotic, and has a role in cell reprogramming. And why we thought it could be a good candidate for research of these habitat-adapted wild genotypes is because it's not only part of the stress response in the plants, but it also plays a central role in defining that stress. And the link between the stress adaptation and these alternative pathway in respiration is really well established. And last but not least, it's highly polymorphic. So we can identify individual genotypes and groups of genotypes within populations. So I thought, okay, let's go wild. And that's what I've structured the program for in 3 steps. First, go to some nice European places, sample for wild carotte, Then go back to the lab, go extract the DNA, try to get my nice sequence of my alternative oxidase, and with some bioinformatical magic, I would have hoped to get a nice model correlating a whole bunch of environmental variables, climaterical variables, soil variables, with my genotypes and my polymorphisms, and hopefully I would find a clade which would relate with a really nice trait. Okay, so that was where I started. So far, so good. I've designed my transect. I thought, okay, let's have a base for that transect would be the Köppen-Geiger climate classification, which is well established, and try to summarize pretty much all the climaterical variables. And I've designed the transect in such a way that it would go through the main classes, the different classes that we have in Southwest Europe. I started sampling end of August, beginning of September, and here I have to say that this study is also going together with the study on mycorrhizal fungal, and therefore, and colonization of mycorrhizal, and therefore I used roots as my target tissue. It's also the target tissue for yield, but I use the roots because I want to use that for also the mycorrhizal analysis. And here was when I started to get problems, and this was about 4 months ago. I was really happy to hear Carlos' presentation today, that Portugal and Spain had a particularly clade. Well, actually, I've designed my primers based on about 70 sequences of AOX1 gene that we have in our lab. They are mainly from inbred lines, cultivars, also some wild crop relatives. And I was actually able, with different sets of primers, to amplify everything that is in that scheme in blue, but I managed no single amplification from the Iberian Peninsula. And that's what I've been struggling in the last 3 months, and that's why I just have kind of conceptual and preliminary data. The only hypothesis that I could put forward is that do we have some sort of biogeographical barrier at the level of the Pyrenees? Because there is clear, at least some different polymorphisms going on there. And Carlos had shown with these, I don't know how many markers, 90 markers, that it forms a separate clade. So I hope next time I can elaborate a bit more on these, and at least there are some differentiation there. Whether it's a true biogeographical barrier, I don't think so, in terms of what we know about, and we've just heard about Daucus invasiveness, but still there is some different evolutionary paths there. Okay, AOX gene, we use it an acid approach, PCR classic, really simple. We get a fragment of around 1,500 base pairs. This is highly variable because of intron 1 size, which on average is about 700 positions, but we got one sequence with just 300 base pairs, so a really short intron, and we got another, for instance, in France, South France, from a wild population in South France with an intron of almost 1,000 base pairs. And actually, this intron, these 350 base pairs more, if you just blast it, it seems that it blasts with Daucus ariensis of Daucus cyrticus, while the rest of the sequences we have blasts totally with Daucus carota. So this is as good as the data that is available on the gene bank. It doesn't say much, but it's nonetheless interesting to recall. It might be a case of hybridization. Some really fast data on the variability of this gene. So the two exons, exon 1 is each something more variable than exon 2. Intron 1 obviously is much more variable. And we try to use different substitution models, obviously, for exons and introns when we try to make our Bayesian likelihood analysis. This is a midpoint rooted tree. And it's, as I said, only for 6 populations instead of the 13 that I would have hoped to be presenting. it doesn't say much. We don't see any clusterings, really. There is two groups, let's say, but the bootstrapping support is about 63%, so I can't take any conclusions out of it. It seems that the population, so it's a bit more blue in that area, but this doesn't say anything to us at this stage. Probably because, first, I do not have enough data. Secondly, because the data corresponds to mainly France, Luxembourg, Netherlands, so this part of the Transact, where there's not that much of rough variation in terms of the classification, except that the population down there, which has a dry summer, actually that's the population weight that appears a bit more separate. But if we put the colors of the code of the climaterical classes, there's not much that I can yet say, except that I hope there's a bit more orange up there, and that with more data this gets a bit more clear. So I know that I'm running out of time, so basically I hope to bring to you soon, and I promise that if I have opportunity next year, I will be presenting a much more complete data set. But I hope that I have soon a bit more of data relating also to this area. This curiosity about hybridization can be as such or not necessarily. My big questions, are there habitat-adapted wild genotypes? Well, I can't say anything now, except maybe. One thing seems clear, if I manage to have all my populations, and the aim is not staying just on this Koppen-Geiger classification, but really go to deep modeling, using all the climaterical variables, to use as well the soil data and so forth, then we hope to have a model that will cluster a couple of populations that, for instance, relate to droughtness or to wet environments or to some specific type of possible stresses. So, yeah, the main borderline that I wanted was that diversity of functional traits is much more relevant than species diversity, I think. But can we find such diversity? And moreover, can we really use it? And the other thing is, I would like, and that was mainly the concept of this talk, instead of going for one size fits all, so just try to go there, pick whatever and see what you have, maybe we can go for a more tailor-made approach, try to really first find that correlation between genotypes and the local environment, and then from then on, go then on much more deep analysis and deep phenotyping, and with much more techniques. Yeah, okay, so basically I wanted to correlate my polymorphisms with soil, weather and climate in much more depth than I've done now, but the main topic also is going to be correlated with the associated mycorrhizal fungal community, which is actually the big scope of the project. And not to be more delayed, I want to thank you. And yeah, this is just the entities that are involved, and this project is done under a Marie Curie Fellowship. And yeah, and I will take any questions. Thank you.
Thank you, Tania. I can confirm that the Pyrenees Mountains are a barrier between France and Spain, but also between France and France.
Okay.
As I said. And also with Corsica. Oh, in Corsica, yeah. Yeah.
Okay. Okay, that is the same sort of...
Good. Any questions? Yes, Shirley, over there.
Hi. So, considering you already have the material and the DNA, have you considered any other genes that might be associated with your climatic data or stress?
Yes. Yes. The point is, well, there's two things that I would actually like to consider, and one of the things I would ask your help, but I've realized now that I'm here that it's much more difficult than I thought, because I would like to have a set of genes that would really try to discriminate. I know that it's Dacus carotte as far as I could. I used a molecular marker, RBCLA, for that, but it's rough. So I would like to, for one, to try to really analyze more in depth at that level. And yes, I have some other genes in mind, in particularly, but they are also related to strongly with the mycorrhizal community, because that's also the other part of the focus. But I'm happy to take any other suggestions.
Well, it's a comment. I'm not a molecular biologist, but you've chosen the alternative oxidase as your and physiologically alternative oxidase is a case of induction in case of the terminal stress induction. So why not use instead something that would be more gradual, like superoxide dismutase?
Okay, that's a good one. For me, there was one clear thing. I wanted something that was not directly linked to any particularly stress, and I wanted to have something drastic. So probably there to go to something that just pops up at a terminal stage, let's call it like that. Because I wanted to try to enhance the chances that I would have to have some sort of correlation at this preliminary analysis between a gene and
superoxide dismutase is very well correlated with water stress.
Yeah. So that's a good tip to take home, also with the suggestion of Shelby. So thanks a lot.